Right, so in today's lesson you have seen part of a whale dissection, and that is to help introduce us to this topic of lipids. I want you to think about, why do whales need to have such an incredible layer of fat? And I do want to encourage you to watch the rest of that video. You can Google it on YouTube. And in fact, there's an incredible series where they dissect a lot of different animals, and those of you zoology fans will particularly enjoy it. Okay, so on the screen I've put what we need to know for this topic, and you need to just go through it and make sure that you can cover all. So you want to go back to these points and make sure that you are confident that you understand those points over there. Okay, so let's go on to lipids. So we've looked at carbohydrates, we're looking at lipids now, and then the next family will be proteins. All lipids, just like carbohydrates, contain carbon, hydrogen and oxygen in their molecules. And some of the important examples of lipids that we are going to study are triglycerides and phospholipids, that should be Li. And they are usually known as fats if they're found in animals, and oils if found in plants. Pause the video to copy this down. Okay, now let's start off by looking at triglycerides. The tri is the bit of the clue. There's something 3 in this, and I want you to picture a capital letter E with its backbone and the 3 lines going down, because that is what a triglyceride molecule looks like. So what is it made up of? The 3 parts is the 3 fatty acid molecules, and it also has 1 glycerol. So the glycerol is the backbone, and the 3 fatty acids make up the horizontal line of the letter E, which is the shape of the triglyceride. Pause, copy this down, and then we'll look at some pictures. Okay, so this is what a glycerol molecule looks like. You can see, because it's going to bond with 3 fatty acids, it only needs 3 carbons in the chain, and it's got 3 hydroxide groups. OH is hydroxide. No need to copy this one down yet. Okay, now the fatty acids, how you know that you're looking at a fatty acid, the big clue is this group at the end, and that is known as a carboxyl group. It's got the C double bond O and the OH. So that is the carboxyl end of the fatty acid. And actually this part of the fatty acid can have all sorts of different combinations, resulting in a lot of variation in the type of fatty acids, and therefore the type of triglycerides you can get. Now, what we need to remember is that when we look at these fatty acids, sometimes, like in this example over here, and you don't need to know the name of the example, but over here we've got some double bonds. Now, you remember from studying alkanes and alkenes last year that the moment you have a double bond, you call it unsaturated. So these over here, this over here is an example of an unsaturated fatty acid, because it's got 1 or more double bond in it. If it's got 1 double bond, we call it a monounsaturated fatty acid, and if it's many, it's a polyunsaturated fatty acids. And that's why often on tubs of margarine they'll say high in polyunsaturated fats. Now, as I said to you, the lipids can also vary, because the fatty acids vary in length, so that's the number of carbons in this long chain here. Different fatty acids, so you can have 3 different fatty acids joining up with the glycerol to make up the triglyceride. And then also some can be saturated, some can be unsaturated, and they could be mono, di or poly unsaturated. Right, so what I'd like you to do, I'm just going to go back for a moment. I'd like you to copy down the glycerol, pause, and then restart and copy down the next slide. Okay, and hopefully you've labeled the hydroxide group. And in the fatty acid, you want to label the carboxyl group. It doesn't matter how many carbons you have here. Pause the video and start when you are ready. Okay. Now you might want to copy some bullet points off here. So pause the video and restart when you are ready. Okay, now I'm sure you can appreciate that because this whole bit here is very, very long and there can be different combinations, and the bit that we are really focused on is this group at the end over here. So for shorthand, I could write it like this. So R means whatever the different combination is. So R means it can be any combination of carbons and hydrogens, but the key part is the carboxyl group, the COOH. Or I could write it like that. Both mean exactly the same thing. So in your notes, just make a note, RCOOH or RC double bond OOH. Okay, so we got the glycerol and the fatty acids, but how do they bond? And this is the bit they love asking. So the key thing is I've got to line up the hydroxides with the carboxyl groups. And just like we did with all our other reactions, we're going to have a condensation reaction where we remove water. So what's going to happen? There we go. The water molecules, so it's the hydroxide from the carboxyl group and the hydrogen from the hydroxide group, are going to be removed, and I'm going to end up with 3 waters. And of course this is known as a condensation reaction. Pause the video and copy this down. Okay, now it's really important. So you've shown step 1, you need to show the final product, and this is the final triglyceride. The bond that you get this time is not a glycosidic bond, but it's known as an ester bond, So the ester bond is shown in red over there. So please make sure you use a different color and label that part as the ester bond. So it says over here, a bond forms between the carboxyl of a fatty acid and the hydroxyl group of the glycerol. A molecule of water is removed to form an ester bond, and this process is called esterification. Pause the video and copy these notes. Okay, you don't need to copy this down, but yeah, you can clearly, if you were given this, you would be able to recognize this is a triglyceride because of the E shape. You can see that this fatty acid is unsaturated, it's monounsaturated. And you can see over here the ester bonds that have formed between the carbon and the oxygen. So here's a question I'd like you to think about. Lipases are enzymes that are involved in the breakdown of triglycerides. Name the bond broken in lipases. And then B, explain how the breakdown of triglycerides would affect the pH of a reaction mixture. Pause the video and answer the questions, and start when you are ready to check your answer. Okay, well done. So the bond broken is of course the ester bond, and the reason why the pH changes is actually the pH starts to go down because the fatty acids are released. Because it's an acid, it lowers the pH, becoming more acidic. Okay? Right, now we've looked at triglycerides. Let's have a look at another example of a lipid, which is the phospholipids. So it says here, sometimes some phosphate ions are found in the cytoplasm. Remember, the cytoplasm is the part of the cell where chemical reactions take place. And sometimes the hydroxyl group in the glycerol undergoes an esterification reaction with the phosphate instead of a fatty acid. So what you have here, there's your phosphate and your glycerol, and you only end up with 2 fatty acids because that third hydroxide has joined with a phosphate group. And in fact, I can draw a big circle around this, both the phosphate and the glycerol group, and that gives you the typical standard picture that we use of a phospholipid. Now, the important thing to remember is this. Where the phosphate group is, the phosphate head, that has a small negative charge, and it makes it soluble. So because it's soluble, it means it dissolves in water. So it's described as a hydrophilic head. Philic means loving, so it's a water-loving head, water-loving part of the molecule. By contrast, the 2 fatty acids form a hydrophobic tail. They'll do anything to stay away from water, and that creates some very interesting things. And we think about very often fat molecules, they stay at the top, they try and repel, move away from the water, and which is why the fat that lands up in a sink full of dirty water doesn't dissolve. So here's how we can draw this in a very simplified way. And you can see over here, at the bottom over here, I've got water, and you can see the phospho heads in the water. So that's the hydrophilic end, and the hydrophobic bit will stick in the air if we have a monolayer. But if I put it in water, because it's surrounded by water, what happens is the little fatty acids, they move to the inside, creating this micelle. This is how this is described, with the phospho heads on the outside. And then with the cell membranes, which we know is a phospholipid bilayer, you've actually got 2 rows of these phospholipid molecules, and the phosphate heads are on the outside. So that means that you've got the aqueous solution, perhaps the cell is near water, or it's near, of course, the cytoplasm, which contains water, or next to another cell. So it makes sense that these that the fatty acid ends are on the inside. Right, getting too excited, let's go back, pause the video, and copy down these notes. Okay, again, pause the video and copy down these notes. Okay, in the next video, I'm going to describe the functions of lipids, both in general and in membranes.